Dear friends, here we are going to provide you clear and detailed description about one more important topic, namely ABC flower model development. This model states how meristematic tissue gets changed into flower. Or we can say it states that plant has well-defined meristematic tissue, how it gets converted into floral meristem, then flower meristem later on forms well-defined flower, or we can say reproductive structure. This model was first formulated by two biologists, namely George Hodgin and Chris Somerville, in plants Arabidopsis thaliana and in one more plant, namely Antiherum majus, or also called a dog flower. This ABC flower model development is genetically controlled process. It involves number of genes. The genes are known as homeotic genes. These genes have three subtypes, or we can say three classes, A, B and class C genes. These genes, they coordinate, or we can say they interact with each other and helps in flower development. Homeotic genes govern the development of anatomical structures in the plant body. Homeotic genes are also present in humans. In humans, homeotic genes help us to develop body plan. For example, head to tail axis. There is a gene known as Hox gene in humans as a homeotic gene. Now coming back to our main topic, let me show you working site of these homeotic genes. Plant has a meristematic tissue. It is mass of undifferentiated cells. It has the highest power of cell division. This meristematic tissue divides in one dimension, forms apical lateral and intercalary meristem on the base of position. Later on, this apical meristem further divides to form ram, or we can say root apical meristem, and well-defined stem, or we can say shoot apical meristem. These homeotic genes, as already discussed, they act on same region, we can say shoot region, shoot apical meristem, and forms flower meristem. Later on, flower meristem continuously divide to form a well-defined reproductive structure called as flower. Now before proceeding, let me show you structure of normal flower, or we can say diagrammatic representation of the flower. Complete flower looks like this. It has outermost flower wall called sepal, it is green and protective wall, then petal, it is mainly colored for pollination. Then there are stamens, we can say male reproductive part, and innermost, very important, this is called carpel, and carpel is considered as female reproductive part. It is when we proceed from outer to inner side of the flower whorls. Let me show some symbols here. This indicates number 1 whorl, this is second whorl, accordingly third. Carpel is considered as whorl number 4. Now let us illustrate how flower organs develop into flower whorls. For this purpose, I am going to show you number of concentric rings. These rings indicate different flower whorls of the flower, as we have already discussed. For example, outermost ring, it is number 1 ring, hope you got, it shows sepal. Second, it shows petal. Third, it shows stamen. And last innermost whorl, it shows carpel. Just remember these symbols. Now let me write it here, types and working of homeotic genes in a detailed manner. As already discussed, these homeotic genes have three subclasses. Class A gene, second one is called class B gene, and third one, very important, this is called class C gene. This class A gene has two subtypes. Number 1 is known as API, means epipetally 1 gene. Second is called AP2, means epipetally 2nd gene. Accordingly, class B genes have two subtypes. Number 1 is called AP3 gene. This is epipetalous 3, and PI means pistillate gene. Now here class C gene, it has only one type, AG gene, means agamer gene. Now these genes interact with each other and control superficial flower whorls in a flower. For example, AP plus AP2, it forms sepals and petals of the flower. Accordingly, AP3 and pistillate gene, it forms petal and we call it stamen of the flower. Now last one, agamer gene, it forms stamen, and last one whorl, this is called carpel. Now to know the concept beautifully, let me show concentric rings once more. So this outermost ring, it has A gene, it expresses to form sepal. Now here second concentric ring, here A interacts with B gene to form petal. Now let me show you third one whorl here and last one whorl, fourth whorl like this. Here on the third whorl, B gene interacts with C to control and form stamen. Now C expresses isolately, individually and form what we called it as corpel of the flower. Now we understood homeotic genes acts as a key gene for flower development. Here we can illustrate working of homeotic genes through one more simple diagrammatic representation. Suppose these are three classes of genes as shown in this simple fig. This is class A gene, this is B class gene and class C genes. It has two subgenes API, epipetalous 1, it gets expressed to form sepals. Now epipetalous 2nd, epipetalous 3rd of B and A gene, they interact and controls what we called as formation of petals. Here pistillate gene, it interacts with agamous gene C and helps to control formation of stamen. Now there lies agamous gene, only C gene individually gets expressed to form corpel. Let me conclude with some important pointers here. Number 1, A gene is antagonistic to gene C. They work independently. B gene cannot work independently. It works when interacts with A or C. Now let me pose question. B gene is common in which two flower development? Right? Answer in comments. Now very interesting point is here. If A mutates, C gene will get expressed. For example, A forms sepal but after mutation it will form corpel. C forms corpel, after mutation it will form sepal. Now let me tell you last part of the story. That is mutational theory of ABC flower model. If all homeotic genes get mutate, what will happen in case number 1? If gene A will get mutate, I will make you here diagram of all three genes like this. You will find if gene A will get mutate, rest of genes means gene B and C, they will remain as such, never get changed. And C gene will get expressed twice. Let me show you it here. This is gene C, this is B gene and this is gene C. Again B, C remain same, C get expressed twice and the mutant flower will be formed like this. Outermost corpel, then these two it will form stamen. It will again form stamen and last one C gene will form what we know as well-defined corpel. So this will be a mutated form if A gene will get mutate. Now let us talk about case number 2nd. It is reverse of first. If gene C will get mutate, the diagram will be like this. I will show you here. The two genes, rest of genes A and B, they will remain as such. And the gene A will get expressed twice. For example, like that. This is gene A, this is gene B, and this is again gene A, right? Now what will be the formation of whorls? It will form, sepals. Again, these two will interact. It will form what we called it as petals. B and A, it interacts and will form petal again. And again, A will individually form what we called as sepal. Now this will be the mutational flower if gene A gets completely mutate. Right, now let us talk about last case number 3rd. That is, if gene B gets mutate, what will be the possibility of new flower whorls in mutated flower? So let me here show you last diagram of this very topic. If gene B gets mutate, only gene A will get expressed. Mind this point. Let me show you here. This is A gene, this is C gene. As B gets mutate, it will remain recessive. Now A will form individually what we called it as sepals. Again A will form here individually. Again same word, this is called a sepal. This is second word. C will get expressed, it will form what we called a corpel. Again same C, it will form corpel. So new mutated flower might have arrangement like sepal, sepal, corpel, corpel from inner to outside. Now if you find this video very informative, we will desperately wait your likes and comments. Thanks, have a nice day.